Hey guys, welcome to my YouTube channel. Last video we'll discuss about the mitosis and the stages of the mitosis, cytokinesis and the significance of the mitosis. So here we'll discuss about the meiosis. Meiosis is also known as the reduction process. The offspring, the production of the offspring by sexual reproduction includes male and female gametes, complete set of the haploid chromosomes. So gametes are formed from the specialized diploid cells. So this specialized kind of the cells division undergoes the reduction. Suppose it is a 2n, it is reduced to n reduction process, production of the haploid daughter cells, and this kind of division is known as the meiosis. Meiosis ensures the production of the haploid phase in the life cycle. Sexually producing organism, fertilization takes place. So it involves the two sequential cycle of the nuclear and cell division called meiosis 1 and the meiosis 2, but only a single cycle of the DNA replication. So meiosis 1 is initiated after parental chromosomes have replicated to produce identical sister chromatids at S phase. In S phase, DNA is doubles. So meiosis involves pairing of the homologous chromosomes and recombination between non-sister chromatids of the homologous chromosomes. 4 haploid cells are formed at the end of the meiosis. For meiosis 1, 2 haploid cells, and after end of the meiosis 2, 4 haploid cells are formed. Meiosis also includes prophase, metaphase, anaphase and telophase. Also meiosis 2 also includes metaphase 2, anaphase 2 and telophase. In meiosis 1, it has several processes. In meiosis 1, prophase 1 is a very largest cycle and a longer, more complex when compared to the prophase of the mitosis. In prophase, it has several stages regarding the chromosomal. It has 5 stages called as the leptotene, zygotene, pachytene, diplotene and dikines. During leptotene stage, chromosomes are gradually visible under the microscope. The compact of the chromosome continues through leptotene. So in the leptotene process, only chromosomes can be seen under the light microscope. So in zygotene, coming to the zygotene process, chromosome starts pairing. So together and called as the synapsis stage, it forms the synapsis is nothing but the pairing of the chromosome. Such chromosomes are called homologous chromosome. Electron micrograph of this stage indicates that chromosome synapsis is accomplished by the formation of the complex structure called synaptic complex. Pairing of the chromosomes is known as synapsis, and the formation of the complex is known as the synaptomal complex. So the complex formed by the pair of the synaptid homologous is known as the bivalent or tetrad. However, they can be seen in next stages very visibly. So here leptotene, zygotene and then pachytene. During pachytene, the 4 chromatids of each bivalent chromosome distinct and clearly appeared as the tetrad. Chromosome condensed and forms the bivalent and shows the tetrad. Tetra means 4 chromatids. This stage is characterized by the appearance of the recombination nodules. When 2 chromosomes are pairing, recombinations may appear. Crossing over takes place between non-sister chromatids of the homologous chromosome. This is a chromosome. This is also a chromosome. These two are non-sister chromatids. So crossing over is exchange of the genetic material between two homologous chromosomes, and it contains the enzyme mediated process is involved known as the recombinase enzyme. Recombinase enzyme it includes. It leads to combination of genetic material of the chromosomes between homolog. At the end of the packaging, chromosome linked to the crossing over the site. And then coming to the diplotin, dissolution means separate of the synaptal complex and tendency to the recombinant homologous of the bivalent to separate from each other. Here the separation will takes place between the synaptal complex and bivalent will separate from each other except at the crossing over. This is important. And it forms the X shape in nature. And then the final stage is known as the dikinesis. It is the termination of the chiasmata. Terminate means to end or eliminate. Chromosomes are fully condensed and meiotic spindle assembled to prepare the homologous chromosomes for the separation. Takes place for the separation. By the end of the dikinesis, nuclear disappears and nuclear also breaks down. It represents the transition of the metaphase. This is all about the meiosis 1. First stage is known as leptotin. Zygotin, pachytin, diplotin, and dikinesis. So this sequence may be asked like the flow chart 1, 2, 3. This is very important. In leptotin, chromosomes can be visible, and in zygotin, pairing takes place is known as the synapsis, and complex formed is the synaptimal complex. So bivalent is seen. As in pachytin, pairing of the synapses of the homologous chromosome, it is known as the bivalent. In diplotin, resolution of the means removing of the chromosomes and it forms a X-shaped in dikinesis, termination of the chiasmata or X-shaped and leads to the separation of the homologous chromosome. At the end of the dikinesis, nucleolus and nuclear envelope all are disappears. This is all about the meiosis 1. Mitosis 1 prophase is very short lived, whereas compared to the meiosis 1, prophase is very longer because of the 5 stages like leptotin, zygotin, pachytin, diplotin and dikinesis. This is all about the meiosis 1. In further video, we'll discuss about the metaphase 1 and 2. If you like this video, please like, share and subscribe. Thank you for watching.